In this series of figures, we want to look at the absorption of fats across the wall of the small intestine. And digestion of fats actually begins earlier in the stomach with the secretion of gastric lipase, as well as previous to that, the secretion of salivary lipase. And that digestion occurs in the stomach, and then that chyme would enter the small intestine, the duodenum, where we further increase that digestion through the secretion of pancreatic lipases. Absorption of those fats would then take place in the latter sections of the small intestine. Now, if we blow up those latter section portions of the small intestine, look at the inside layer of that wall, we see that it's lined by surface area adaptations, one of which is the villi. If we take one of those villi and blow it up here, we can see its overall structure. Now we see that it's lined by the cells of the mucosa, known as the enterocytes. In the small intestine, they're actually lined with microvilli, further increasing the surface area for absorption of nutrients. And on the interstitial side of that lining of enterocytes, we can see that we have capillaries. These are fenestrated capillaries with relatively high permeability. That's going to allow nutrients that are being absorbed across this enterocyte lining and into that bloodstream. Now, this would only include the solutes, the nutrients that are permeable across those capillary walls. So essentially, the carbohydrates, the monosaccharides, and the amino acids. And that's because they're small and hydrophilic. The lipophilic fats need to be coated and solubilized by certain substances along the way as they move through their processing here. And they cannot enter, they're called chylomicrons here. We'll see we form those in a little bit here. But those guys cannot enter the capillaries due to their protein coating. And so they have to enter a lymphatic vessel. And that lymphatic vessel will carry those solubilized fats back to the circulatory system through lymphatic ducts. Now, if we take a section of these enterocytes, just a few of them, and blow them up over here on the right, we can look at how the fats are processed and packaged for their absorption across that wall. Now, here we are in the lumen of that small intestine. We've got our fats partially digested. A primary source of fat in our diet would be triglycerides. We also eat cholesterol and phospholipids. Those would also be absorbed. And our pancreatic lipase is acting to digest those triglycerides, break them up into their component-free fatty acids and the leftover monoglycerides here. And those fats need to be repackaged and delivered to the wall of the small intestine here for absorption. Now remember, the fats are lipophilic. They're going to tend to cluster together into these big droplets, and so we have to emulsify them. That's where these bile salts came in. Those were produced by the liver and released from the gallbladder in response to the food in the small intestine. So we have our fats being solubilized by the bile salts, but they need further breakdown. And so our pancreatic lipase here needs to access those fats to continue their breakdown. Well, the lipase molecule is hydrophilic, and our bile salts sort of oppose their association with those fats, right? The bile salts are emulsifying the fats, but it actually creates an inhibition for this lipase. And so we actually need a helper. We have colipase that tethers the lipase to this micelle, this bile salt-coated fat droplet, keeping that lipase associated with it and allowing for that digestion to continue. Now that micelle can approach the enterocyte lining here, and we see that we're going to protonate some of those fats. We can get this micelle close enough that the fats inside this micelle become protonated by this acidic environment, and those are taken up into the membrane here, right? They're lipids, and so they dissolve into the lipid membranes of these cells, but then have to be removed by this fatty acid binding protein. This allows us to deliver them into the aqueous cytoplasm for further processing. Now the cholesterol and the phospholipids are part of what have been taken up across here. Those are going to be repackaged down here. We're going to resynthesize triglycerides. The triglycerides, as you can see here, can't actually enter directly. They get repelled, right? And so we have to break them down, get them across the wall, and then resynthesize those triglycerides. And then we're essentially repackaging these fat droplets. But in this case, we're protein coating them. We're going to use an apolipoprotein here, and we're going to form a chylomicron. So that's a protein-coated fat droplet. Whereas out here, a micelle was a much larger bile salt-coated fat droplet. These chylomicrons are exocytosed onto the interstitial side. Once again, they can't enter the capillaries, right? They're coated in proteins. They're too big to enter the capillaries due to that low permeability to these large bodies, these large emulsified fat droplets. But they are permeable into the lymphatic vessels. They're called lacteals. And those will lead us back into the central circulatory system where they can be added to the bloodstream.